Hi, my name is Erica Nagy, and today I will be touching upon the phases of mitosis and the important events occurring within each stage. So mitosis is the process of cell division and replication that occurs in our somatic cells within our bodies. Our sex cells or our gametes undergo a similar process called meiosis, but today we'll really just be focusing on mitosis. Mitosis is the process of DNA replication and division, and it results in two daughter cells, both of which are diploid and identical to both each other and the parent cell. The first stage of mitosis is interphase. So in interphase, we have the G1 phase of mitosis, and this is a period of DNA growth. And the DNA is growing in a sense that it's synthesizing the required molecules, the required proteins that it will need to continue on with the replication and the division of the cell. Following G1, the cell may either continue to the S phase of mitosis or pause in the G0 stage. The S phase of mitosis is where we see DNA undergoing replication in preparation for cell division. After the S phase, we have the G2 phase, which, similar to the G1 phase, sees a period of cell growth and DNA preparation for the rest of mitosis. After the three stages of interphase occur, we have prophase. In prophase, spindle fiber apparati, or centrosomes, will start to form, and these will later aid in separation of our chromosomes. The DNA will condense into sister chromatids, which are two chromosomes that are conjoined at the centromere. And the nuclear envelope, which originally is surrounding our DNA, begins to dissipate, which will allow the spindle fibers ultimately to attach to the centromere and aid in the separation of those sister chromatids. Following prophase is prometaphase, and in prometaphase, the nuclear envelope continues to break down. The spindle fibers are now able to continue migrating to opposite poles of the cell, and they begin to attach to the centromeres as well. Here we start to see DNA begin to align along the equatorial plane. It's not exactly at that equator yet, but it's starting to line up and migrate. Prometaphase leads the cell into metaphase, and this is when sister chromatids are aligned right on the equatorial plate of the cell with the centrosomes at both opposite poles, and this kind of allows the cell to line up in preparation for the chromatids to be separated from one another. After metaphase, we see anaphase, and during anaphase, the spindle fibers begin to shorten, and it pulls these sister chromatids apart. So each chromosome of the sister chromatid is being pulled to opposite poles of the cell, and this will set the cell up to divide into two identical daughter cells. So telophase and cytokinesis are the final stages of mitosis. In telophase, the spindle fibers will start to dissipate, and we'll see the nuclear envelope start to reform around the two new sets of DNA on opposite poles of the parent cell. Cytokinesis is kind of distinct from all the other stages of mitosis. Up until this point, the cell has been duplicating within the parent cell, but when cytokinesis occurs, the parent cell divides into two daughter cells. So all of that genetic material and the two sets of separated DNA that are at opposite poles will now be divided into their own daughter cells. In plant cells, cytokinesis occurs when a cell plate is formed right down the equatorial line of the cell. And in animals, the cell will start to pinch off, and it forms what we call a cleavage furrow. And again, this will produce two identical daughter cells, both with two N chromosomes, both diploid in nature, and both identical to each other and the parent cell as well.